Students, this is Professor R.M. Mahindrakar from JSS Arts, Science and Commerce College, Gokak, who is welcoming you for this new session on gene ecology or ecological genetics of population. Species in nature differ in their environmental requirements. They also differ in respect to their tolerance to the environmental fluctuations. The species occur in several morphological forms in different habitat conditions. Goethe Thoresson, in the year 1922, a Swedish worker, conducted a series of experiments on variations within a Swedish plant species, Plantago maritima, whose image is towards the right. He collected a group of 20 or more individuals of the same species from different areas and planted them under the identical environmental conditions in his experimental garden at Akrak. He noted that several integrated forms existed within the species and they differed from one another in morphological and physiological features. Some of the differences might be unstable and induced only by the environmental factors, while some others might be permanent. That is, they may be genetically induced. These observations led him to formulate the concept of gene ecology, which brought about many revolutionary changes in the plant taxonomy. Thoresson observation based on population sampling and subsequent procedures led him to conclude that species differ from one another morphologically, physiologically, and in habit requirement. On the basis of such studies, he proposed the following classes of morphological forms within a species. These are the ecads, ecopenes or habit forms, ecotypes, Ecoclines, Ecospecies, and Quinospecies. Let's start with ecads or ecopenes. These are also called as IP hormones or habitat forms. These are environmental variations. They belong to the same genetic stock or species, and the variations in their morphology, that is in shape, size, number, and reproductive capacity, are induced by environmental influences. The variations are not fixed, but are temporary, somatic, and reversible. If one type of ecad is transplanted into the environment of another type of ecad, the differences would disappear. The morphological variations are not permanent because the genetic composition of ecad is not affected, and so they are identical in their genetic behavior and interfertile. That is, they can be crossed with each other. The variants of ecopenes may differ to such an extent that they can be treated as a separate species. Let us see some of the examples of ecads. Euphorbia hirta plant, when this plant is growing in the grassland, they are prostrate and profusely branched, while the plant of the same species growing on footpath are compact, small-leaved and cushioned. When these forms are grown under identical conditions, the differences disappear. Now, this we can observe in front of our college library. Both the forms of ecads are available in the open air theatre. Now, if you see the green variety, that is the prostrate variety, you will see it growing near the trees under the shade. And along the footpath, you can see that the Euphorbia hirta, its ecad, is showing such a condition here. Now, this image is actually from our college ground itself. Now, another example is in this Dichanthium caricosum. Several distinct morphological forms of ecads have been noted under different habitat by Pandey in these plants. In both the species, there are two types of ecads: basket form habitat by the plant growing in protected areas, which you can see towards the left side. That is, this image shows the basket form, and saucer shaped habitat of the same plant is seen in those areas where overgrazing is there. This is because grazing causes reduction in the size of the erect stem, number of spikes per raceme, number of spikelets per spike, and in length and breadth of lower glumes of spikelet. In addition to these morphological variations, some physiological variations are also noted, as for the example of development of anthocyanin in the leaves and early initiation of flowers. Now the next is ecotype. These are also called ecological or physiological races. The term ecotype was proposed by Thoresen in the year 1922 to the grouping of populations or ecological races or subspecies of a species in relation to different environmental or habitat conditions. These are interfertile forms or biotypes of a species which possess different genetic compositions or genotype and arise due to mutation, hybridization, and isolation. That is, when these two different ecotypes are crossed, fertile offsprings are produced. But here they are genetically different. There will be a slight variation in their genetic composition, unlike the ecads. In ecads, there is no genetic differences, but in ecotype, there is genetic differences. In ecads, they are also interfertile. Ecotypes are also ecofertile. Though the different ecotypes of a species are morphologically and genetically distinct, yet because of their interfertility, they are put into one taxonomic species. Ecotypes are morphologically, physiologically, and developmentally adapted to live under varying environmental conditions. Ecotype is a product of genetic response of a population to a habitat. In ecotype, adaptations are irreversible. That is, they retain their features even when planted in the neutral habitat. The difference in the ecotypes are so marked that some of them are treated as separate species by old taxonomists. Mishra and Shiva Rao observed ecotypes in the species Ledenbergia polyalthea. Ramakrishnan has shown that red and green population of Euphorbia thymifolia are the two ecotypes. And Shiva Rao in the year 1948 found Ledenbergia polyantha and Ledenbergia utricifolia to be ecotypes of the same species. Now here the image that you're seeing, this is an image of this Ledenbergia polyantha. Now this form of ecotype grows in those areas which is rich in lime, whereas the Ledenbergia utricifolia, this is a ecotype of the same plant species, that is the same genus, but this grows in those areas where there is less amount of lime. Similarly, Ramakrishnan in the year 1959 has shown the red and green population of Euphorbia thymifolia as the two ecotypes. Now this is an example of a green Euphorbia thymifolia, which grows in those areas where the calcium content is less. So it is a calcifuge. Whereas the red ecotype of this Euphorbia thymifolia. It grows in those areas where the calcium content is more. So it is a calcicole. Ecotypes have been reported in several plant species such as Euphorbia hirta, then Cassia tora, Argentina quinqueris, then Sencus ciliaris. During the last 2 decades, many investigations have studied either only a few populations or several widely separated populations and have shown that each population was an ecotype, and the term is now being widely used below the level of species. Our increasing knowledge of gene ecology and population differentiation has shown that ecotypes may be the ecological unit but not an evolutionary unit. Now let us see how these ecotypes originate in nature. There are three methods by which the ecotypes might arise in nature. The first is by hybridization, that is natural hybridization. It is produced by natural cross between two species. For example, when Spartia stricta is naturally crossed with this Spartia ultimifolia, a new hybrid, Spartia townsendi, resulted. Now here towards the left, this is Spartia stricta. Towards the extreme right is the Spartia ultimifolia, and in the middle we have this a new hybrid Spartia townsendi. Now this eliminated both the parents from their natural habitat because it is having greater adaptability than the parents. The second method is mutation. Mutation are the sudden changes in the gene composition. Now these natural mutations and recombination in small gene pools accumulated in a segregating population, which makes it better adapted to the particular habitat or environment. Some new ecotypes also arise by cultivation or protected growth as it eliminates competitive selection. The third method is by chromosomal change. Structural change in the chromosome, such as translocation, inversion, and loss or addition of chromosome segment, produce change in genotype and phenotype, resulting in formation of a new ecotype. Polyploidy also leads to the formation of new ecotypes because polyploidy hardly exhibit ecological tolerance as their parents do. Now we shall see the different kinds of ecotypes. Ecotypes have been observed in large number of species, and the cause for ecotype differentiation may be the altitude, latitude, light, soil, biotic interference, physiological changes, etc. According to the varying environmental conditions, ecotypes may be of following types. Climatic ecotype. Ecotypes which are produced due to varying climatic factor as light, temperature, water, and wind are called climatic ecotypes. Thureson has recorded the climatic ecotypes in this Leontodon autumnalis. Now here you can see in this image, there are three different types of the ecotypes of Leontodon autumnalis, which are growing in different environmental conditions, and that is why you see they have their different physiological changes. We can see different morphological changes. You can even see the structure also changes in them, and these changes are genetically fixed. The next is edapic ecotypes. Ecotypes which are produced due to differences in edapic or soil factors. such as soil moisture, excess of or deficiency of nutrients, change of soil pH, etc. All these come under the edaphic ecotypes. Mishra and Rao have studied Lyrenbergia polyantha, and Krishnan has studied Euphorbia thymifolia and recorded several ecotypes in them. Now these examples were already seen earlier. Now the third is this climatic edaphic ecotype. The ecotype produced due to the influence of both the climatic and edaphic factors are called climatic edaphic ecotypes. Pandey and Jayant in the year 1970 have reported climatic edaphic ecotypes in this Cenchrus ciliaris. Now these are the plants of that, this is the image of that plant, Cenchrus ciliaris. Now both are growing in different climatic conditions and different edaphic conditions. So that is why you see the change in their morphological structures, and you can see how they have altered themselves and suited to meet their present conditions. The next is altitudinal and latitudinal ecotypes. The ecotypes are produced due to change in altitude and latitude. Such ecotypes are found in Cassiatora, Angagalis, Pinus, and many other gymnosperms. Now here you can see these are the 4 ecotypes of Cassiatora. Even in Gokak also we have one species of Cassiatora. Now this is the species of Cassiatora we can see in this Karbagatti forest. Along the roadside you can see those yellow colored plant, yellow flowering plants, and also you can see this variety also in the Karbagatti plant. So these two ecotypes are seen in our Gokak climate. And these are all because of variations in their altitude and latitude conditions. So that is why they are called as altitudinal and latitudinal ecotypes. Next is this physiological ecotypes. These ecotypes are produced due to physiological changes as in the photoperiod, water absorption, nutrition uptake, etc. For example, this Boutilona curtipendula. Now, this variety is a long day plant, this variety is a short day plant. I mean, this ecotype is a long day plant and this ecotype is a short day plant. So, the physiological change, because here the plant has modified itself so that the flowering occurs in short day conditions, and this has modified itself so that the flowering occurs in the long day conditions. So these are both are the ecotypes of the species Boutilona curtipendula. Now we shall see some of the basic characteristic features of these ecotypes. Ecotype of a species, though genetically distinct, are always interfertile. That is, they can be bred with each other so that you get fertile offsprings. They retain their original features when cultivated in neutral habitat. That is, one plant growing in lower altitude and another plant growing in higher altitude. When you plant both those plants in the same altitude, they will be retaining their original morphological features. That is, the plant growing in the lower altitude will retain its same features, and the plant growing in higher altitude also will retain its same features even if they are grown in the same environmental conditions. Ecotypes are genetically fixed. That is, one ecotype is different in their genetic composition from the other, but they are all interfertile. They, the differences in the characters of ecotypes are actually due to natural selection or locally adapted populations. That is here the whatever the variations we see, that is due to the adaptations. Now we shall see some of the significance of ecotypes. Cultivation of economically important plants has been made possible in different habitats. New ecotypes of a species enable it to be adapted to climatically and edapically different places. Ecotype help the species to extend its ecological range and spread to new area. Morphological variations may be marked in the species growing on varying habitat, which leads to evolution. Third morphological forms are these ecoclines. Ecoclines are the intermediate forms which are found in the transitional regions between two adjacent regions occupied by separate ecotype of a species. They are produced by hybridization between two related ecotypes, and they survive under intermediate environmental conditions. Thus, a continuous change in the characteristics of the plant from one region to another region is apparent. The ecocline usually describes the ecotone in which a series of communities display a continuous gradient. It is the gradual or continuous change of an ecosystem or community. A wetland would be a good example of an ecocline in that it is a gradual transition from a terrestrial ecosystem to a aquatic one. Now here, these are the terrestrial plants. Now in the wetland, what happens is these forms, they get accustomed to the new environment, that is the aquatic adaptations. So they develop certain characters. So these transitional stages where mesophyte is converting its morphological features, anatomical features into a hydrophyte. So these transitional forms, we call them as these ecoclines. Now here there is a diagrammatic representation of how this coniferous forest is getting adapted to this aquatic medium through these transitional stages, that is ecoclines. Now the fourth morphological forms are these ecospecies. According to Greger, ecospecies is a unit of gene ecological classification. An ecospecies has one or more ecotypes, but ecotypes of one ecospecies do not produce viable offsprings when crossed with the ecotype of another ecospecies. That is, a cross between two ecospecies never produce fertile offsprings. Example, if two ecotones, an ecotone describes an area that acts as a transition or boundary between two ecosystems, has been separated from one another to a very long time, the adaptations become a permanent part of the gene. They are still the same species, but their differences physically and genetically are very distinct. In fact, two ecospecies cannot produce viable offsprings when they are crossed with each other. These ecospecies, when left alone for many generations, then develop sufficient changes in them to become a separate species. That's the long version of how new species are born. What happens if the environmental conditions exceed the ecological amplitude? extinction. The current extinction crisis is because of this very reason. The organisms are unable to adapt quickly because their amplitude is limited with respect to the changing environment conditions. Now here there are four ecospecies of this plant, the Phycomithila patens. What do we mean by this coinospecies? Different ecospecies which can undergo occasional crossing but do not produce viable offsprings are collectively brought under coinospecies. In other words, populations incapable of exchanging genes with other populations of the same species are referred to as cynospecies. So my dear students, here I conclude this topic on gene ecology. So now whenever you are outside, please try to observe the different types of ecads and ecospecies around you and try to learn. And if you find any new species, bring it to the college. So let us investigate it. And please take care of your health, wear mask whenever you go outside, and be safe. Thank you very much.